Okay, so now officially welcome everyone. Very nice to see you here in today's session. Today's session will be hosted by me, Hannah Broeks, and Laura Pape. We are both PhD students within the BioClock Consortium, which organizes these sessions. So the BioClock Consortium is a Dutch research organization which focuses on studying different aspects of the biological clock in healthcare, in society, and in nature, so very broad. And the aim of this lecture series is to equip you with some basic concepts and provide you with knowledge from real experts in the fields of varying sides of chronobiology. So just some practical stuff. This seminar series is organized every third Wednesday of the month. It will be approximately a 40-minute talk and then 50 minutes of discussion. Usually we do it after, but I heard already that there will be an opportunity also for you to ask questions in the middle as a pause for everyone. But during the talk, you can just also post your questions in the chat so we can see if there are any urgent matters. Yeah, I think that's the general stuff. So then I give the floor to Laura, who will introduce you to our speaker.
Yes, thank you, Hannah. Today, we are very happy to welcome Dr. Hassan Dashti, who is an assistant professor of anesthesia, critical care, and pain medicine at Massachusetts General Research Institute and assistant professor of anesthesia at Harvard Medical School. He is a registered dietitian and researcher with interest in clinical nutrition, chronobiology, sleep, and genetics. And his lab focuses on clinical and translational research on sleep and circadian rhythms in patients with nutrition-related challenges. And he aims to develop personalized medical treatments for diverse patient groups. And in his talk today, he will inform us about the role of food timing in cardiometabolic disease risk factors and the future directions of chrononutrition. Yeah, we are very happy to have you here, and we give the floor to you.
Great. Hello, everyone. Thank you, Laura, and thank you, Hannah, for the introduction. Thank you all for being here. It's nice to see so many of you and also some names that I recognize. I appreciate you taking the time to be here, especially at the end of your workday. As Laura had mentioned, the title of my talk today is Advancing Chrononutrition for Cardiometabolic Health, with an emphasis on opportunities for future research. So the talk will primarily be split into two parts, knowing that this is a diverse crowd with various interests and backgrounds in chrononutrition. I'm going to start more broadly introducing to you the concepts of chrononutrition and its impact on cardiometabolic health based off existing literature. We're going to take some break. Maybe I'll take a couple of questions if they're highly relevant to what I've presented on. The second part, I'll be talking about or highlighting future research opportunities in chrononutrition. And hopefully I'll inspire you to find ways to integrate chrononutrition assessments into your own research projects. Maybe not as a primary focus, but as a secondary or an additional focus to your work. Okay, so with that, I will get started. So as I mentioned, I'll start with an introduction to chrononutrition to get everyone here up to speed. This is by no means a comprehensive review of the topic, obviously because of the limited time that we have. But what I wanted to do is really highlight the different types of studies that have been done in the field over time and demonstrate how did we get to the point that we're at today. Okay. So traditionally, when we talk about nutrition in general, we overall emphasize quantity and quality of foods. So when we eat, what we eat and how much we eat. Those have predominantly been the two themes that constitute the major dietary guidelines or guidelines that tell us how we should be eating. So what we eat reflects the quality of our food. There's an emphasis, for example, on eating foods that are that tend to be healthier or have higher healthy eating index, food patterns that are higher in healthy eating index, foods that are more plant-based rather than animal-based. That's an example of the quality dimension. There's a lot of emphasis on how much we should be eating, so the amounts of calories that we should be consuming, grams of sugar, grams of fiber, as an example. However, an emerging player in the field is timing, which will primarily be the emphasis of today. However, those dimensions are not necessarily separate. There's some overlap. Looking at them in combination is probably the best way to go. So I'm just trying to move a couple of things on my screen. Okay. There's no accepted definition yet for chrononutrition. However, we recently had a workshop a few years ago where we attempted to put together a definition for chrononutrition, which I'm presenting on the screen. Chrononutrition is undoubtedly an emerging dimension of nutrition. It's an active area of research that examines how timing-related aspects of nutrition impacts circadian rhythms, biological processes, the pathogenesis of disease, and also its potential as a form of chronotherapy. So it's really not only the impact of timing on health, but also its impact on biology, as well as its potential or its promise as a therapeutic once we understand that biology and its connection with human health and disease. Luckily, I don't need to explain to this crowd what circadian rhythms are and what the biological clock is. However, the only thing that I think that I want to reiterate and remind you all is that just like how light is an important synchronizer of circadian rhythms, food and exercise are also additional environmental cues that influences or synchronizes the organization of circadian clocks, primarily clocks in peripheral tissues. So food intake as well as exercise could influence the rhythmicity of clocks in peripheral tissues, including the liver and muscles, and less so the central clock, which is primarily the effects of, rely on the effects of light exposure. And I mentioned this to really highlight that there's really an established biological basis of why timing of food intake is highly relevant. It's because eating at the wrong times could induce two things that could lead to impaired health. First, eating at the wrong times could induce internal desynchrony, and then its potential in inducing circadian misalignment. So because food is an potent environmental cue, is an important Zeitgeber, just like light exposure, it has the capability of synchronizing the human biology to the external environment. However, it primarily affects the programming or the reprogramming of clocks in peripheral tissues. So eating at times where we do not necessarily expect to be eating could lead to this internal desynchrony or internal desynchronization across various clocks within an organism. So by only influencing clocks in the periphery, but not the central clocks, that leads to a state of internal desynchrony, which we attribute to some of the detrimental effects of eating at the wrong times. The other effect that could explain why eating at the wrong times is detrimental is its potential in inducing circadian misalignment. So we know that key biological processes and metabolic processes are optimized at certain times of the day, typically during the biological morning, and they are impaired during the biological evening. So what happens when we eat at the wrong times is that we induce a state of circadian misalignment where our bodies are not really optimized to metabolize and digest foods, and this suboptimal state results in circadian misalignment, which has been linked to impaired cardiometabolic health. So mismatch between our behaviors as well as our circadian cycles, which primarily happens when we eat at night or during our biological evening, results in circadian misalignment, which along with internal desynchrony could lead to all these impaired cardiometabolic health outcomes. Okay. And I summarize these 2 points to you to really demonstrate that there's really a strong biological basis of why food timing or chrononutrition is an important aspect of nutrition to consider in nutrition research as well as in biomedical research in general. Okay, but as I've mentioned very early on, chrononutrition is an emerging field of nutrition. It hasn't been studied as long as some other fields that have been studied. And I want to take you through a series of work that have been published over several decades and just showcase to you some important studies that have been done generally in the field. And actually, the first mention of chrononutrition doesn't emerge in the literature, at least on PubMed, until recently in 2007. But there have been studies prior to 2007 that have considered the timing of nutrient intake. And the first set of studies that have been done looking at some timing aspects of nutrition were really done on studying the composition of breast milk. The first study that I was able to identify was a Lancet study, actually published in 1949, many decades ago, that compared the content of fats in various samples of breast milk collected across a 24-hour clock. So what they did was analyze fat content in breast milk collected at different times of the day and plot how the fat composition varies across 24 hours. This study, along with several other studies done around the same time, were able to demonstrate that the 24-hour composition of breast milk changes or differs across the clock, and morning milk, or morning human milk, appears to be elevated in various proteins and fats that are responsible in promoting activity, whereas they are elevated for other fats and other proteins that could promote inactivity or sedentary or sleep behaviors. And this makes sense. The mother is signaling, the parent is signaling to the infant to be active during the daytime, whereas to sleep during the nighttime. And I showed you in that plot earlier that the first mention of chrononutrition, at least on PubMed, appeared in 2007. And actually, those first couple of studies in 2007 all the way to 2009 were all related to breast milk and its varying composition across 24 hours of the day. It wasn't until the late 70s when we start seeing some aspect of cardiometabolic health now emerging for chrononutrition and linking the timing of food intake with various cardiometabolic outcomes. And those first set of studies were conducted actually by Army labs or military labs here in the US. And you might wonder, why would the military be interested in chrononutrition? And that's basically because of the erratic food intake of various Army personnel. As you know, food might be limited during in war, for example. What the Army was interested in is whether breakfast or dinner should be prioritized if rations were very limited. So in order to answer that question, the Army labs, this was an Army lab actually in Massachusetts, they did this comprehensive study with a really nice study design. They recruited healthy volunteers here, and they restricted their food intake to only morning. So they had all the participants consume their calories as breakfast, so in the morning, or as dinner in the evening. What they did really nicely is have a 24-hour physiological measurements done throughout the day. And what they were able to show was that eating dinner rather than breakfast led to shifts in various cardiometabolic parameters, including shifts in the rhythmicity of blood pressure, temperature, and circulating levels of immune cells. And what they were able to conclude is that restricting eating to the evening led to, again, phase changes in the rhythmicity of various metabolic processes, including the secretion of growth hormones and insulin. And now you're starting to see a tie-in of chrononutrition with cardiometabolic outcomes, which started with these types of studies led by the Army labs. With the turn of the century, we start seeing now links between food timing and weight trajectory. And the first study that I will highlight is an animal study where, and it's a highly cited study, very simple study design, where what the investigators did was provide a high-fat meal to mice, either exclusively during the day or exclusively during the night. So keeping in mind that mice are nocturnal animals, it's natural to them to be eating during the dark and not during the light. What they were able to show in this 6-week experiment was that the mice being fed this high-fat meal during the light gained significantly more weight compared to the mice that were being fed exclusively during the dark, which is the right time that they typically eat. You start seeing the differentiation in weight changes between the two groups as early as week 1, and that the weight change persists over the 6-week experiment. There's roughly a 5-gram difference between those two groups of mice. And again, this is independent of how much high-fat calories were being consumed by the mice, or obviously the source of the calories. This effect is exclusively attributed to the effects of when the calories are being consumed. Now, this led to a lot of interest in trying to translate these findings to humans. Does the timing of food intake affect weight trajectories in humans? Does it affect weight loss interventions, the efficacy of weight loss interventions? One of the most cited studies in the field was a secondary analysis of a weight loss intervention, which was published in 2013. It's a highly referenced study. What they did in this secondary analysis was stratify weight loss participants enrolled in this behavioral weight loss intervention into early and late eaters. And this was a 20-week behavioral weight loss intervention. And what they were able to demonstrate was that participants who were enrolled in this weight loss intervention, who were eating earlier, were able to lose more weight in response to their intervention compared to those who were eating at a later time. In this study, participants were stratified based off of their lunch time. So early eaters were those who were eating lunch before 3 p.m. Late eaters were those who were eating lunch after 3 p.m. And this is a Spanish study, which explains why they selected those late cutoffs for early and late eating. But you can see very clearly that the late eaters were losing less weight versus the early eaters. And this effect, again, is independent of the sources of the calories and the amount of calories. So this 2-kilogram difference that they identified, that they saw at the end of the weight loss intervention, is entirely attributed to this timing effect. And in addition to this study, there were a lot of other similar epidemiological studies that did the secondary analysis to examine the effects of timing on weight loss as well as on weight. A big question that later emerged is, should we now be paying attention to the timing of food intake relative to our biological clock, as suggested or implied by the animal studies that I've presented, or is a societal clock more relevant, as might have been suggested by this behavioral intervention that I presented on earlier? And to answer that question is this study published in 2017, where they enrolled lean and non-lean adults and compared the timing of their food intake relative to their DILMOs, so their biological night, as well as a separate comparison relative to societal clock, or basically to clock times. And what they were able to show, as can be seen here in panel A, is that the lean and non-lean individuals, you see clear discrimination between the two when you stratify them relative to their DILMOs. What they were able to demonstrate was that non-lean individuals, or more obese individuals, were eating closer to their DILMOs, roughly 1.1 hours closer to the onset of their biological evening, relative to the non-lean individuals. Whereas when you start comparing them relative to societal clock, you see an entire overlap between the lean and the non-lean individuals. What this demonstrates is that perhaps it's the biological clock eating relative to the biological evening rather than to a fixed societal clock time might be more relevant as a cardiometabolic for cardiometabolic health outcomes. So we really need to be focusing on biological evening rather than eating relative to our 24-hour clocks, societal clock. A few more studies to highlight. There have been a lot of work done on night shift workers because of their behaviors that happen at the wrong times of day. So it's not only that they're active during the biological evening and expose themselves to the biological evening, but also because their food intake primarily occurs during the evening whenever they conduct these night shifts. One of the studies that I have been involved in this field is this one, where we looked at the associations between night shifts and type 2 diabetes. And this was an analysis done in the UK Biobank, where we were able to look at the effects of night shifts in over 160,000 individuals, which included night shift workers as well as day workers. And what we were able to show in this epidemiological study was that compared to day workers, conducting night shift work was associated with a higher risk of type 2 diabetes, and also that the magnitude of this association was exacerbated the more night shift work that you do. So people who did more night shifts had a higher risk of having type 2 diabetes compared to those who were doing fewer night shift work. And even when we adjust for BMI, you can see an attenuation of that association, but the association still persists, which tells us that this effect is not entirely because of weight, but rather there are these other behaviors that could explain why we're seeing this risk, which could partly be attributed to eating at night, as well as light exposure at night, as well as exercise during the night as well. Okay. Now, most of the studies that I've shown you so far have been primarily epidemiological, with a couple of exceptions. But now in the 2020s, we start getting more money in the field. We get more robust studies being conducted. And I'll highlight two studies that were published in 2022. The first one is a randomized crossover clinical trial, which enrolled 845 individuals, many of whom were overweight and obese. And what they wanted to do with this crossover trial is compare the effect of an oral glucose tolerance test 4 hours before bedtime, which simulated an early dinner, comparing against a late dinner timing, which was scheduled an hour before bedtime. So 2 oral glucose tolerance tests, an early dinner timing test, and a late dinner timing test. And these were the results from the oral glucose tolerance test. What I'm showing first are the glucose results at the 5 time points that were measured during this oral glucose tolerance test: time point 0, 30, 60, 90, and 120. On the y-axis, we have the glucose in milligrams per deciliter. The dark lines are the late condition, so the late eating, whereas the bright gray line is the early eating. And you can see huge glucose intolerance during the late compared to the early condition. And what's important to note here is that, again, this is in response to the same glucose load. So a 75-gram glucose, you can see a huge attenuation in glucose tolerance when the glucose is consumed during the late condition compared to the early condition. And what this study was also able to do was measure insulin as well. And what they were able to show through the series of insulin measurements was an attenuation in insulin secretion at the 30 and 60 and somewhat in the 90-minute time points, which suggests that the body's ability to respond to the glucose bolus is delayed, and it takes longer for insulin to get secreted in the night, in the late eating condition compared to the early eating condition. A lot of this effect is directly attributed to the effects of melatonin, which partly what it does is impair insulin secretion from beta pancreatic cells. What this study was also able to do was now look at the effects of genetics in addition to late eating to see whether there are specific predisposed individuals based off of their genetic profiles who should be more cautious about late eating. The specific variant that this study was interested in was a common type 2 diabetes risk variant, a gene called the melatonin receptor 1b gene, which is known to be associated with higher type 2 diabetes risk. And what they were able to show, and as demonstrated in these plots, is that the effects of late eating is actually more detrimental in those who carry the diabetes risk variant. So you can see that the delta, the difference between the two curves here is larger. The difference is larger in those who are carrying the diabetes risk, the GG genotype, compared to those who carry the wild type or the non-diabetes risk variant, the CC genotype. Which really suggests that while late eating compared to early eating is detrimental for everyone, it's particularly relevant and more detrimental and harmful for those who carry specific variants, including type 2 diabetes variants, in specifically the MTNR1B genetic variant, which is the type 2 diabetes risk gene. In addition to the glucose effects, they were able to show some insulin effects in that the insulin secretion defect or impairment is larger in those who carry the defective form or the risk form of the gene. This is one of the two studies in 2022 that were published. The second one, led by Dr. Frank Scheer, also had a very nice crossover study design where they were able to carefully shift the timing of meals from early schedules to late schedules. And to be brief, what they were able to show was that shifting meals from earlier to later in this crossover study design was associated with a reduction in energy expenditure. So there was a reduction in metabolic rate, a reduction in core body temperature. There was an increase in hunger as well. And through various metabolomics analysis, they were able to pinpoint exactly which pathways were involved to explain these behaviors or to explain these results. So they, for example, see a reduction in pathways related to autophagy, whereas an increase in in the expression of pathways related to lipid anabolism. So now with these two studies, you start seeing more robust study design, these crossover study design, but you also see a layering of omics. In the first study, we see genetics being introduced now in studies on chrononutrition. And now in this cell metabolism study, we see metabolomics being introduced in the study of chrononutrition. In 2023, the NIH, the National Institutes of Health here in the US, started recognizing the importance of chrononutrition as an understudied, but possibly a very important aspect of nutrition. And what the National Institute of Heart, Lung, Blood Institutes in the US did was organize a 2-day online symposium to highlight chrononutrition. And you might wonder, why is the NIH interested in this field? As I said, they were interested because they started recognizing it as a potential modifiable behavior that could promote cardiometabolic health once we understand exactly how it is related to cardiometabolic health. They recognize that there are a lot of gaps and barriers in the field that needs to be addressed in order to effectively advance the field. And also, chrononutrition is aligned with many priorities of the National Institutes of Health here in the US, not only those that are relevant to nutrition, but also ones that are relevant to sleep. So as a result, there was a 2-day workshop that I was involved in. It was a virtual workshop. Maybe some of you have attended this, that discussed the state of the field of chrononutrition. And it's really not just the scientific community that is very interested in chrononutrition, but it's also the public. People love the idea of how food timing could have influenced their health. And only last week, there was a really nice article published in the New York Times. And I'm showing you a screenshot of that article that the title of the article was "Should Dinner Be the Smallest Meal of the Day?" And I really like this this picture that was published alongside the article that shows breakfast, lunch, and dinner using 3 different sizes of plates, with breakfast being the largest plate, lunch being middle in size, and then dinner being the smallest size plate, to emphasize that what we eat or when we eat is probably just as important factor for nutrition as what and how much we eat. Although I will argue that this small dinner plate is probably exaggerated. A lot of the foods that appear on this plate are pretty good, so I don't think it necessarily needs to be that small. But what I wanted to showcase here is that it's not really just the scientific community that's interested in food timing, but it's also the general public that is really interested in this idea of when we eat might be relevant for overall health. Okay, so this is a timeline that I wanted to present. Hopefully this gives you all a glimpse and a snapshot of time of different studies that have been done, different study designs maybe that have been included in various studies. And also, this is a turning point where I'll start now talking about what are future research opportunities in chrononutrition. So, as it has become obvious, there are a lot of possibilities and a lot of gaps in the literature. Obviously, not what I shared is not comprehensive, and it's in no way includes all the studies that have been conducted. One of the aims of the NHLBI workshop was to highlight some opportunities and gaps in the field, hopefully to inspire researchers to integrate into their future studies. And that's what I'll be doing next. I'll be highlighting some of these key themes and critical gaps that have been discussed. I'll spend more time on some of these rather than others. This is a figure in the upcoming report from the workshop, which hopefully will be out shortly. Okay. And I'll just start going through all 8 points, and I'll spend more time on some rather than others. So if you think of it, just like there are different ways to think of quality of food and quantity of food, there are different ways to think of timing. And I've kind of alluded to that throughout my presentation. What isn't clear yet is what are all the possible dimensions of chrononutrition and which ones are most relevant to cardiometabolic health and general health in human. And I'm going to show you just different ways that we can think of chrononutrition, going from the simplest to the more complex way. And a lot of these are actually also related to the ways that we might be thinking of sleep as well. A lot of this is inspired by the sleep community. The simplest form of thinking of chrononutrition is thinking about the timing of various meals or the timing of the first and the last meals. So just the clock times. This is meant to show a 24-hour clock. Thinking of the timing of your meals, your breakfast, lunch, and dinner, first and last meal, is probably the simplest form of chrononutrition that you can study. You could then, from these, start looking at other aspects of chrononutrition, such as the duration of your daily eating window by looking at the delta in time from the time you start and stop eating, and also the duration of your overnight fast and the timing of that fast as well. These are all examples of looking at timing relative to societal clock, but now we could look at timing relative to our sleep schedules. And perhaps this is more biologically relevant because we start integrating the biological evening. So rather than thinking of timing relative to societal clock, we can now start thinking of when do we start eating relative to wake time? When do we stop eating relative to sleep time? And how much of this might be coinciding or the proximity of this to our sleep episodes. So far, we've considered meals as we've considered meals as a binary variable. However, we know that we eat different meals have different calories. So layering the calories on top of the timing adds another layer of complexity. So rather than just looking at the timing of the meals relative to societal clock and sleep-wake cycles, how about considering, in addition, the amounts of calories that are being consumed during those different meals? If our dinner is the smallest and our breakfast is the biggest, does that have the same effect as switching or having breakfast being the smallest and dinner being the largest? Another layer of complexity is if we start looking at the macronutrient composition. So not just the calories, but what are the sources of those calories during those different times of day? Does that matter for cardiometabolic health? And then probably the most complex thing that you can do is now looking at variability across days of these various compositions. So this goes to show you that there are different variables of chrononutrition and also different layers of complexity. And a lot of this will be affected by how you've measured timing-related variables in your study. A major limitation in the field, partly because it is a newer field, is having those standardized terminology. And we really need to standardize terminology so that when we talk about chrononutrition, we're all on the same page. We all know what we mean. For example, what is late eating? A lot of people talk about late eating. Many studies reference late eating. However, there's no standard definition for late eating. When people say late eating, do they mean late eating relative to biological clock, relative to sleep, relative to societal clock? People and even in pop culture, people talk about eating at the right time. You should eat at the right time. What does that mean? Are they talking about biological time, societal time? So we really need to standardize the terminology in order to then create those guidelines on how do we assess eating times relative or based off of these standard terms. An area of interest is also generating these frameworks that integrate not only a single aspect or a single variable of chrononutrition, but integrating many of these concepts in chrononutrition. And again, this is somewhat borrowed from the sleep community, where they have this nice integrated Are You Sated sleep framework that includes various components of sleep, including regularity, satisfaction, alertness, timing, efficiency, and duration. So can we do the same thing for chrononutrition? Not only looking at the timing, but also eating window, variability across days, and also integrating the calories and the quality in addition to the timing. So this is an area of huge interest and an area of opportunity, creating these frameworks. In order to do those, we obviously need established and robust tools to measure food timing. What I'll be doing over the next couple of slides is just take you through some of these existing tools that are available out there. Some of those have been validated. Some of those are newer and need more time in order to validate and replicate. The simplest way to measure food timing is obviously through single questions. You can ask participants, for example, in surveys, at what time did you have breakfast, lunch, and dinner? People have gone about to modify accepted tools in nutrition in order to integrate an aspect of timing. For example, this is a screenshot of a food record where we ask participants to write down what they eat and how much they eat. We added this timing column in order to ask them now to report also at what times they ate. There have been a couple of meal pattern questionnaires, so questionnaires that are focusing predominantly on timing. And I'll show you a couple of those in a second, but this is a screenshot of one of those that exists. And then just like every other field, there's an interest in using technology in order to objectively measure food timing. And I'll mention a couple of those opportunities as well. We did some work in order to validate survey questions for food timing. What we did is come up with these very simple food timing questions, asking participants to recall what time did you first start eating, stop eating, and when did you have your main meal. We compared those responses against prospectively collected food timing in food records to see what is the correlation or the concordance between recalled versus prospectively collected food timing. And we found really nice correlation between recall and prospectively collected food timing for the first and the last meals. However, people don't necessarily are able to recall the timing of their main meal, which could be because people don't know how to classify main meal. Is it main meal relative to the calories or main meal relative to the amount of time you dedicate to having that meal? So asking people about their first and last meal could be a good way to characterize meal timing. But asking about the main meal could be a bit problematic because of inability of people to really classify main meals. I mentioned that there are a lot of questionnaires actively being developed. Some of these have been validated and replicated. Some of them still need some time for replication. So if you want simple questionnaires, these are some examples. I wanted to highlight one of them, which is this meal pattern questionnaire. What I like about this specific questionnaire is that it doesn't ask people what they ate. People hate recording what they ate, and they have a really hard time recalling how much they ate. This questionnaire only asks about timing. It asks people to jot down what are the times they usually eat, and whether at those times do they have a main meal, a light meal, a snack, or a drink. So very simple way to capture temporality of calories. Technology is being used for food timing research. We did this really cool study where we used cafeteria. This is a picture of our cafeterias here at the hospital. I was interested in the timing of when employees were eating. We used the timestamps on the purchases from the cafeterias to estimate food timing. Obviously, there's a lot of assumptions being done here, and that people eat exactly at the same time that they buy their foods, which is not necessarily always the case. But we actually saw in the study in that early chronotypes, so early birds, were purchasing earlier than later chronotypes or late owls, which somewhat validates this approach. But what I wanted to highlight is that you can think outside the box with timing. A lot of things are time-stamped, and that gives you an opportunity to study food timing. A lot of mobile applications, this is Meal Logger, also include timestamps, which allows you to look at timing. So whereas participants might log in what they ate, the automatic timestamps also captures timing, which is nice. There's a huge interest in using continuous glucose monitoring for chrononutrition research. So what CGMs do is measure interstitial glucose every 5 or every 15 minutes, depending on the model, over 24 hours. And by looking at this CGM plot or this glucose plot, you can see that perhaps we can estimate food timing episodes based off of these curves. We know that there's a spike in glucose every time we eat something that has some carbohydrates. And there have been some algorithms that have been developed, although not yet replicated, that determine food timing based off of this novel technology, which is now being used not only in patients with diabetes, but also some other populations as well. Okay, so just a quick summary. We need to put a lot of work in developing some of these assessment tools. There have been efforts to modify existing nutrition tools to assess food timing, such as the ASA24, which is commonly used as a food record or food diary. There's an interest in wearable technology, maybe using some omics to estimate food timing, and then obviously to integrate multiple aspects of health in order to assess food timing as well. Time-restricted eating. So time-restricted eating is not the same as chrononutrition because it primarily focuses on the length of your eating and your fasting windows with little to no attention to when these eating windows happen. Some studies have now started to layer timing to time-restricted eating so that interventions are focused on not only the timing eating window, but also when do these eating fasting windows should happen. So we're starting to seeing a layering of time-restricted eating or intermittent fasting with chrononutrition, and this is still an area of huge research and research that is emerging, whereas most of the prior research in time-restricted eating has focused predominantly on just the length of these windows rather than the timing of these windows. There's a huge emphasis and interest in studying vulnerable populations. I mentioned already why night shift workers are considered a vulnerable population. We do know very well that conducting any night shift work disrupts the body's rhythms, and we know very well that these individuals are at an increased risk not only for cardiovascular disease, but also for other diseases, including cancer. And there's an interest in trying to develop these food timing interventions in order to counter the effects of shift work. We know we will forever need people to do night shifts because they are important to the workforce. So we need to probably invest more time and effort to understand, okay, how could we counter the detrimental cardiometabolic effects that result from shift work? And it could potentially be through these food timing interventions. So this idea of chrononutrition as a chronotherapeutic. Okay, food timing, again, is not the same as the timing of physical activity. However, the two are somewhat related. And there's an interest, again, in including both timing of nutrition and timing of physical activity, looking at those side by side in order to advance the field. And just moving just a little bit more quickly to leave more time for a Q&A. Just like how there's no one-size-fits-all for general nutrition recommendations. We also think that chrononutrition effects likely differ from individual to individual. I showed you those effects vary depending on genetics. I gave you one example of the melatonin receptor 1B gene, but perhaps those recommendations could further be focused or tailored based off of age groups, sex, genetic chronotype, so whether you're a morning or an evening person, and overall health status. Okay. I will skip this one slide for the interest of time in order to wrap up. The last slide here is really recognizing that the time we eat is determined by a lot of factors. There are social influences, behavioral influences, and contextual influences that affect when we choose to eat. Today's a work day here or everywhere. I'll only be able to eat during my lunch hour. So work affects when we eat. And it's very important to understand what are all these influencers in order to come up with the best interventions or chrononutrition related interventions. We list here a couple of determinants of food timing. Things like sex and age are important factors that influence when people eat as well. Okay. Just a very quick summary, standardized terminologies, assessment tools. We need more robust and validated tools. We need to consider effect modifiers, and we need a deeper understanding of social, behavioral, cultural influencers of chrononutrition, food timing, in order to advance the field of chrononutrition, and especially in order to develop interventions for chrononutrition. Okay. I'd like to thank a lot of people who have supported my research over the years, as well as the NHLBI. And with that, I will end there. And I see a couple of questions in the chat. And if I don't get to your questions, feel free to reach out to me after this call. And thank you for listening.